Hello and welcome to an overview of Chapter 13. This is going to be a pretty long one and a doozy, so sit down, buckle up, and let's go for a ride. These two scientists are Watson and Crick, who are often credited with the discovery of the DNA structure, or discovering what the structure of DNA was. As we will discuss later, it was not just their work that contributed to us understanding that DNA is the inheritable genetic material that codes for proteins and traits and phenotypes. So last chapter we talked about Morgan and the fruit fly experiments that he was doing, and at this point in the scientific community it was well established that there were chromosomes in cells because we could visibly see them under microscopes, but we were not sure which of these two materials, DNA or protein, was the candidate for genetic material. We had a pretty good idea of what proteins were based on experiments, but there was this unknown factor of what actually was the composition of chromosomes within the nuclei of cells. So one experiment that was really important was Griffith's transformational experiments with bacteria. And he did experiments with different strains or varieties of a bacterium species, meaning that they had different genetic traits. We would know that now. He wasn't aware of that, but he did know that they had different phenotypes. One was pathogenic, meaning it was lethal, and one was harmless. And what he found is when he mixed heat-killed remains of the pathogenic strain with living cells of the harmless strain, some of those living cells became pathogenic. And his experiment is summed up pretty well with this image where living cells that were the control, these were the pathogenic cells. If they were then given to a host cell, that host would die. The non-pathogenic cell given to a host wouldn't kill the host. Heat-killed S cells led to the mouse being healthy, but then if there was a mixture of the heat-killed S cells and the non-pathogenic cells, some sort of transformation occurred where then some of the living cells became pathogenic and the mouse died. And we would now understand this to be the horizontal gene transfer that can occur between bacteria, which allows bacteria to obtain new genetic information from their environment. But at the same time, this was good evidence against proteins because we were denaturing the proteins when we killed them or when we heated them up, because proteins' tertiary structure becomes unwound when it is heated up. Now later, Oswald Avery also did some work that saw that DNA was indeed the transformational substance. And what was happening in the scientific community was that many biologists remained skeptical because they didn't really know about the structure of DNA, the composition, and the idea was that proteins ought to be the better candidate for genetic material. And this was revolutionized when we discovered viruses. Now, viruses, as we are very well aware with COVID, are non-living vessels of genetic material encapsulated in proteins. And while we didn't know this at the time, we were able to see viruses and see their effects on plants and organisms. So in 1952, Alfred Hershey and Martha Chase did work with DNA, and they used it to understand, or they understood DNA through the work of a virus known as a phage, known as T2. And phages are viruses that infect bacteria. And to determine what they did is they designed an experiment showing that not only DNA of the T2 phage and not the protein enters E. coli, and this they used to conclude that injected DNA provided the genetic material for proteins. So what they did is they grew viruses in radioactive sulfur and radioactive phosphorus. And they did this because sulfur is predominantly found in proteins, and phosphorus, they knew at the time, was found in some sort of genetic material. And then they let the bacteria infect the cells. They then put the bacteria in centrifuges, basically to separate the different constituent parts, the different molecules of the bacteria, and what they found was the radioactivity had not entered the bacteria yet, but remained in the liquids. But however, the sulfur, or I should say the phosphorus, was found in the pellet or the bacteria solids, which means that the proteins had obtained material from the phosphorus and not the sulfur, which was giving us some good evidence that maybe proteins weren't this genetic material. It was also known that DNA was a polymer of nucleotides, so we were getting more information that indeed nucleotides and not amino acids were the primary building block of DNA, but we still didn't have an idea of what that DNA looked like or what its structure was. And in 1950, Shagraph found out that DNA composition, the A's, the T's, the G's, and the C's, all varied from one species to the next in terms of their cellular composition. He is well known for the Shagraph ratio, that A's and T's plus G's and C's equals 100% of all nucleotides. So we did start to discover that indeed the structure of a nucleotide has three parts. There is a phosphate molecule, which gives DNA a very negatively charged strand. There's a 5-carbon or pentose sugar with a 5, 4, 3, 2, and 1 sugar placement. The 5 and the 3 are really important for discussing DNA's composition. And then finally, a nitrogenous base. A nitrogenous base might have 1 or 2 rings depending on the nucleotide that we focus in on. And there are 4 nucleotides that we should be familiar with. There's guanine and cytosine and adenine and thymine. Adenine and guanine are purines. They have 1, 2 nitrogenous bases. And cytosine and thymine are pyrimidines. They have 1 nitrogenous base. You will notice that DNA is sort of synthesized in a 5' to 3' fashion. So we read DNA as TACG rather than GCAT. Now, the 2 findings became known as Shagraph's rule. Basically, the base composition of DNA is obviously different between species. And if you have the value of A, that is going to be the value of T. And if you have the value of G, that's going to be the value of C. Now, Rosalind Franklin, who was an early chemist who started her work in the coal industry and then worked on the tobacco mosaic virus, was working at a lab in Cambridge in the 1950s, and her work was done with X-ray diffraction. So she would shoot X-rays through crystals containing substances and then be able to look at those X-ray scatterings to understand the structure of a molecule. And her work found that DNA was indeed a double helix structure with 2 distinct strands that were complementary to each other, and that roughly the width of a DNA strand was 1 nanometer. Actually, this is the radius, not the diameter, and that the spiral was about 3.4 nanometers for 1 complete turn. Now, some evidence that we now know about to understand the structure of the DNA is that we need to have complementary base pairs based on the size of nitrogen bonding, or nitrogen base pairs. 2 pyrimidines will result in a width that is too narrow. 2 purines will result in a DNA strand that's too wide. A purine and a pyrimidine gives us that nice 1 nanometer width. This is also really helpful during the S phase of mitosis to make sure that there aren't any errors, because enzymes can use active sites to determine whether or not the strand is too wide or too narrow, and then have fixes occur to prevent errors being inherited in new cells. And that's not the only way that we can have error checks within DNA structure. We now know that adenine and thymine have 1,2 hydrogen bonds. Remember, these are intermolecular forces based on negative and positive interactions between molecules. They aren't sharing electrons. There's no electrons gained or lost. This is just sort of temporarily interactions of attraction based on the charges of different atoms. And alternatively, G and C have 1, 2, 3 hydrogen bonds based on those negative positive interactions. I always remember 2 because thymine has the letter T, just like 2, and C and 3 rhyme, which helps me remember GC3. That might be a clever way for you to remember those differences. Thymine goes 2, GC3. And this is another way for us to determine errors within DNA because a G or a C that only has 2 hydrogen bonds because they're bound to an A or a T will have that sort of unbound active region where an electron group is quite electronegative, and enzymes can sense that and help us determine errors within the structure of DNA. Now, the next question was really, how is DNA even replicated? And there was a couple of hypotheses as to what was going on within the structure of DNA as it was replicated. There was the idea that there was the conservative model, which basically, if we have a double-stranded DNA, cells are going to do something where the parent strand remains intact, and there's a complete new daughter strand. There's a semi-conservative model which said that each strand will separate and then code for a complementary strand. And then there's the dispersive model, the idea that sort of like we're just going to chop up the DNA in different regions and we're going to have a mixture of new and parental strands in all of these. And to sort of test this, what happened is we cultured bacteria in some heavy isotope nitrogen-15. And we then allowed that bacteria to grow and obtain the nitrogen because this is going to be incorporated into, all of our nitrogenous bases. We then took all of that bacteria after growth had occurred and put them in some lighter nitrogen, some isotopes of nitrogen-14 that weren't radioactive, and then we allowed growth to occur again. And the results were quite interesting. After the first replication, we had a very noticeable, just simple band. And then after a second replication, we saw that there were distinct bands of more and less dense. Now, why do these results sort of help us with the semi-conservative model? Well, it points to the fact that over time, new template strands that we added to parental strands are going to separate and then code for more nitrogen-14. Well, the initial parental strands that were grown in nitrogen-15 will remain towards the bottom of the tube, and over time this band will get larger and larger and larger as more and more growth occurs in the lighter 15. The dispersive model would have worked for the first time when there was a mixture, but then the argument would be that we would have lots of different bands all within one large section because they would all contain 14 and 15. So the separation points to the fact that over time, each strand becomes complementary to a new strand over replication. So what happens is the parental molecule splits, they separate, and then they form new complementary strands. And then if this would happen again, each of these would be the parental strand for the next replication. Now, getting started with DNA replication begins at what we call the origin of replication. And this is going to be a location with a predetermined sequence that allows DNA to be separated, which allows us to have what's called a replication bubble. At each end of a bubble is a replication fork where we see a Y-shaped region occur. And this is where the parental strands are being unwound to allow new complementary daughter strands or new strands to be formed. And this is what we sort of see at a replication fork. This is the intact parental strand. This is one strand and a second strand of the parental strands that are going to allow us to have new complementary strands occurring. So there's a lot of enzymes that we need to talk about in this image in terms of what's going on with DNA replication. The first is one of my favorite bio names ever. It's Topoisomerase. And Topoisomerase, its job is to sort of stabilize the DNA strand and unwind the tightness that occurs as we unwind this. So as we unwind DNA, by simple shear force, the strand further down the replication fork is going to sort of twist and wind because of tightness, and this job is to sort of unravel or loosen all of that pressure. Helicase is going to be the enzyme that sort of unwinds the hydrogen bond, so it's going to be unzipping the zipper or undoing the Velcro, so to speak, of DNA. And then primase or RNA primer is going to be added to strands to sequence complementary strands in a 5-to-3 fashion. There are single-stranded binding proteins that often bind to parts of the DNA to help with primase or DNA polymerase to come to the party and also stabilize the DNA strand as it is working. And this then can be seen in both eukaryotic and prokaryotic cells, where over time and at the origin of replication, we will have daughter strands coming in both directions, which sort of creates a circle within a circle. And as we create two circles within each other, we'll get to the point where the two circles divide, and this helps cause bacteria to grow and divide quite quickly without the use of centrioles or kinetochores or microtubules, because the two circular DNA chromosomes push each other apart, helping split the cytosol. At the same time, eukaryotic cells have chromosomes that are linear, so we often see lots of replication forks occurring simultaneously, where all of these enzymes are duplicating two chromosomes to make, or one chromosome to make two chromosomes simultaneously. This is useful because we have a lot more base pairs in our genetic material than just a simple bacteria. We have 23 pairs of chromosomes, and bacteria only has one circular chromosome. Now, an interesting thing to note is that DNA polymerases, the enzymes that actually create new strands of DNA, can't actually initiate synthesis until there is a primer, sort of like an initial coat of paint. So we're going to be using that RNA primase to add a short initial nucleotide strand of RNA nucleotides that will soon be replaced afterwards. So what often happens is we add a little bit of RNA with RNA primase, and then DNA polymerase can come in and start synthesizing a long strand of DNA. There is a difference between the leading and lagging strand, and that has to do with the fact that we can only synthesize DNA in a 5-to-3' direction. So if we're synthesizing DNA in 5-to-3 here, that means that this DNA strand is 3-to-5. And the same is true for the top parental strand. More on that in a second. So you can sort of see with our parental DNA that we have a 5-to-3' DNA strand up here, which is why we can add a little bit of RNA primer here and then just have DNA polymerase come in and do a continuous, nice long strand of DNA until replication finishes. Here's our helicase. Now at the opposite end of the spectrum, we'll be talking about lagging strands in a second. We can sort of see how this sort of moves like a zipper or a zipper, whatever is on a zipper that unzips it, sort of moves down the zipper, and also the single-stranded proteins also do that as well. Now, so what happens is we have our RNA primer makes the primase makes the RNA primer, and then DNA polymerase 3 is going to add a little bit of information. Now this is happening for what we call Okazaki fragments or lagging strands, and we'll talk about why this occurs in a second. And then once we get back to the origin of replication, the fork, we're going to have to redo this. So what's happening in the lagging strands is that if we have DNA that is being synthesized in a 5-to-3 direction, which means that this is 3-to-5, we can't just synthesize a long strand because we're going away from the replication fork. So what's going to be happening is we unzip a little bit with our helicase, we add some RNA primer, and then we synthesize DNA. We unzip a little bit more, add some more RNA primer, do some more DNA. This isn't a problem for the other strand that's being built on the bottom because this is being synthesized in a 5 to 3' direction continuously because this strand is 3 to 5 and this strand is 5 to 3. Alternatively, the same thing is true for this bottom strand, this top leading strand here. It's being synthesized in 5 to 3 because that is the direction of the synthesis, but it can just continuously follow the helicase, drafting it as it unwinds. So with our Okazaki fragments, we're going to have this continuous mismatch of DNA polymerase 3 adding some nucleotides and then unzipping the origin of replication a little bit more, adding some RNA primase, and then having some more DNA polymerase. Now, what happens after DNA synthesis occurs, or replication, is that DNA polymerase 1 is going to come in and replace RNA with DNA to make those lagging strands, those Okazaki fragments, a little bit more intact. They're going to be getting rid of those RNA primers, and then DNA ligase, remember ligand is a bond, comes in and sort of stitches the pieces together to make one continuous fragment. And this is what we finally get with our overall strand. So we have our leading strand up here, which is just following along with helicase. And this is because if the top strand is 5 to 3, we can go 5 to 3 on the leading strand. Alternatively, the bottom parental strand is 3 to 5 prime. So that means we have to be synthesizing in this direction, 5 to 3, which is why we have to continuously add primers as we unzip our zipper. Now, you might be saying, wait, is there a big difference in the time between the leading and lagging? Not really, because enzymes are so efficient with their jobs based on their protein structure. There's really no difference between a leading and lagging strand in terms of efficiency and time. It's just that we need more actors to do the removing of the primers and the tying together of the Okazaki fragments. Now, this can happen simultaneously many times in eukaryotic cells, which is why we tend to have sort of this complex structure. And this generally has many connecting proteins attaching to different parts of the DNA synthesis complex, which allows us to be doing multiple parts of the DNA at the same time in an efficient manner. Now, DNA can be damaged by exposure to harmful chemicals. There's a whole list of things under the sun that can cause cancer or cause DNA damage or mutations. There's also physical agents such as cigarettes and x-rays. We call these carcinogenic, and this can cause big changes in our DNA composition. We call the ability to repair excision, where a nuclease can actually go in and replace damaged stretches of DNA when an error is detected. So let's say we have an error where we might have some non-complementary base pairs. The nuclease, which is an enzyme, can recognize that there is a mismatch in either hydrogen bonds or purines and pyrimidines. It can cut it out. DNA polymerase can come back in, and then DNA ligase can replicate. So we have a nuclease, sort of like an exorcism of the bad DNA, the repair by DNA polymerase, and then the stitching up by DNA ligase. Now, if chromosomes, something to note right here is that we have all of these chromosomes that are highlighted in fluorescent. And what we've what we've tagged in fluorescence are things called telomeres. And as a chromosome ages and undergoes replication, essential genes could eventually be missing because of damage or the inability to duplicate, which is why we have this enzyme called telomerase, which sort of causes the telomeres, which we'll talk about in a minute, to be lengthened in all of our germ cells or our somatic cells, skin cells, liver cells, red blood cells, all of the cells that aren't gametes. And this allows us to have basically ends at each of our chromosomes that are non-coding DNA segments that allow some buffer to occur to protect all of the coding genes within the main part of the chromatids. Now, chromatin is a complex of DNA and proteins. This is why earlier on scientists were a little skeptical or unsure of what actually was within each type of structure. Now, chromatin is just a complex of DNA and protein, and it's found in every part of our eukaryotic cells within the nucleus. Now, chromosomes fit into the nucleus through an elaborate multi-level system of packing. There are meters of DNA in each cell, yet a cell is only a few nanometers. So that means we need to do some pretty elaborate folding and packing, origami on steroids, you could say, that allow us to fit all of that genetic material in a nucleus. But also, there's huge differences in how that chromatin is packaged and organized throughout the course of a cell cycle. So we start off with our DNA structure, which is just a 2 nanometer diameter or 1 nanometer width. And because DNA is super negatively charged, we use a protein that sort of looks like a lollipop called a histone to interact with it. And histones have a lot of positively charged amino acids in their tertiary structure, which allows for a really neat packing. And histones sort of form a complex called H1. You can see 1, 2, 3, 4, 5, 6, 7, 8 of them that form sort of this association together that wrap DNA around in a coil. And that's going to cause DNA to condense 5-fold in terms of their diameter. So we're going to go from 2 nanometers to 10 nanometers, and we call this a nucleosome. But then we pack all those nucleosomes into 30 nanometer fibers. So we've now made the diameter of DNA 15 times more condensed, where we're sort of putting all of the histones together. We call this chromatid euchromatin, so it's really condensed. And then we take the 30 nanometers and we add some scaffolding proteins that make it 300 nanometer fiber. And that 300 nanometer fiber is then packaged into 700 nanometer chromatids. And we now know that replicated chromosomes, hello mitosis, if we have one chromatid at 700 nanometers, the replicated chromosomes are going to be 1400 nanometers, which means that we've effectively reduced the length of DNA by 350 times through the use of some clever protein interactions. Then this allows us to have easy duplication and division during mitosis and meiosis without losing any genetic material in the move. And this is going to be what generally DNA looks like only during meiosis and mitosis. The rest of the time, DNA is going to be a little less formal, a little less structured, and it's going to look something like this, which allows for easier gene expression and duplication of DNA during S phase. Now, many bacteria contain plasmids, which are just small circular DNA molecules that actually replicate separately from bacteria chromosomes. And this is really useful because we can actually genetically engineer bacteria to make proteins for us. So to clone pieces of DNA, researchers first obtain a plasmid, and they insert DNA from another source, foreign DNA, into it. This resulting plasmid is called recombinant DNA. So here's our bacteria with our nice circular bacteria chromosome, and here's the plasmid, which is a small circular segment of DNA. We could have gotten this, or the bacteria could have gotten this from maybe a virus or from another bacteria through genetic swapping, and we take some DNA of maybe a human or a banana, and we insert it into that plasmid. We then put the plasmid back into the bacteria cell, and if you remember our E. coli lesson, E. coli grow like gangbusters, so we have all of a sudden thousands and thousands of copies of that protein or that gene of interest that we can then harvest. And we could use that to maybe make growth hormones or proteins, or have copies of genes that we could use to put into plants for resistance, or we could alter bacteria to clean up toxic waste. The world is basically our oyster when it comes to doing recombinant DNA through bacteria. Because bacteria grow so fast that we can have many copies of the gene or many proteins produced from this. And indeed, this is how we get things like synthetic insulin or growth hormones to use in medical purposes. We used to have to harvest insulin, for example, from pig livers or pancreases, and that wasn't always very viable for diabetics. Now, why can we do this? Bacteria use these things called restriction enzymes that cut DNA molecules at specific DNA sequences called restriction sites. Sort of like if you've ever cut something out, you follow the pattern. These restriction enzymes see very specific segments of DNA and they cut them out. A restriction enzyme usually makes many cuts, resulting in many restriction fragments, and that allows us to then stick things together. So let's say this is our DNA plasmid in a bacteria. Here's our restriction site. Well, we're going to add a restriction enzyme that recognizes this site to cut it in half. We have generally things called sticky ends, which is a protrusion of one, not two strands of DNA. We also have a restriction enzyme that cuts DNA from a foreign substance at that same very site. And that allows us to then sort of insert the recombinant DNA, the foreign DNA, into that hole within the plasmid, and now we have a recombinant DNA molecule. DNA ligase is going to come in and seal the strands. And to see those fragments produced by DNA cutting, we can use a technology called gel electrophoresis, which allows us to see a mixture of nucleic acid fragments based on length. So we can cut DNA at certain restriction sites and see what the resulting lengths are. And this allows us to compare different samples of DNA to see matches between different samples, because each of these fragments from two sources is going to be of the same length, which is good evidence that there might be some linkage. We can also see some overlap here, some overlap here. So when we go to compare different samples of DNA, we're looking for the overlaps. And what's happening with gel electrophoresis is we add samples of DNA to wells, and then we turn on a positive charge at the right side of the electrophoresis, and the negatively charged DNA molecules, think phosphate groups, are going to migrate across the well. And the smaller the segment, the faster it's going to make itself through the gel. Now we can also use PCR to sort of obtain many copies of a gene or a restriction fragment using hydrogen bonds and some fun enzymes. So PCR can make many copies of a specific target segment of DNA, and it's a 3-step cycle that allows us to have exponential growth with identical DNA molecules. What happens is we need to use a heat-stable DNA polymerase called Tac Polymerase to make this occur. So what happens is we put in a target sequence of DNA and we denature it with heat, about 130 degrees Fahrenheit. Now, you will know that 130 degrees Fahrenheit would kill you if you had that internal body temperature. So we use Tac Polymerase, which is an enzyme found in bacteria in hot water springs, to add the primers. We then cool the DNA and allow DNA polymerase to do DNA synthesis, and that allows us to have 2 copies of an initial sequence. And then we can repeat, so 2 to 4, 4 to 8, so on and so forth, where we denature with heat, anneal with Tac Polymerase, and then extend by cooling and allowing DNA polymerase 3 and 1 to do its job. I hope this has been helpful. I know that chapter 13 is a really long chapter, so please see me if you have any questions, and we'll be using some of this information after break. Thank you and have a good day.